Last time we talked about getting the genome into the cell. Now I want to go through in the next 5 or 6 lectures all the events that happen to make proteins, make new virus particles, and so forth. And today we're going to start by talking about RNA synthesis for RNA viruses. We're going to look at different kinds of viruses separately. We'll look at DNA viruses, and we'll even look at retroviruses separately. But today, RNA-directed RNA synthesis. So just to give you a little context, you should know when these things roughly happened. You know, yesterday, a million years ago, whenever. 1935, tobacco mosaic virus was crystallized. Remember, that was the first virus to be discovered. So Stanley made crystals. Actually, this happened at the Research Institute in Princeton. And eventually that led to figuring out the structure of the virus. But when you make crystals, you have to have pretty pure material. And he was studying these crystals and found that 5% of the crystal was RNA. But people didn't know what that meant at the time. And since it was so little, they figured it didn't mean anything. In 1945, DNA was shown to be genetic material in bacteria. Avery McLeod-McCarty, the famous experiment. In 1952, the Hershey-Chase experiment, which we talked about, showing that the DNA of a bacteriophage is the genetic material. The structure of DNA was solved in 1953. And in 1956, the nucleic acid of tobacco mosaic virus was shown to be infectious, meaning you put the RNA, the nucleic acid, into a cell and you get viruses out. So it was the first time that RNA was shown to be genetic material. So that was a big deal. We didn't think RNA would be the genetic material because all other microorganisms, all other life on Earth has DNA as its genetic material. By 1959, we found RNA in a lot of different viruses, and then people started to study it, and to this day, we study RNA viruses of all different sorts. They're very important. So today, we're going to talk about RNA synthesis of 3 of the Baltimore classes. This scheme should look familiar with mRNA in the middle. We're going to talk about viruses with plus-strand RNA genomes that can be messenger RNA. We're going to talk about viruses with minus-strand RNA genomes. Minus-strand, of course, is not message. You have to make RNA when these viruses get in cells, and they have to bring in a polymerase with them. And we're going to talk about the viruses with double-stranded RNA genomes. The first RNA polymerase in a virus-infected cell was discovered right here in New York City in the 1960s at the Rockefeller Institute. David Baltimore, whose name you've heard, the Baltimore scheme. He was a graduate student at Rockefeller University, and he was trying to figure out how RNA viruses replicated. Interestingly, the prevailing notion was that RNA viruses had to go through a DNA intermediate because no one knew of any enzymes that could copy RNA into RNA. So Baltimore was doing an experiment. This was a PhD thesis experiment of his. He was infecting cells with poliovirus, and then at different hours after infection, he would break open the cells and make a cell-free extract. And then he would add radioactive nucleoside triphosphates. And then the polymerases in the extract, whether they were viral or cellular, would take up the triphosphates and incorporate it into nucleic acid. And so this is a graph of his experiment. Here's virus production, plaque-forming units per mil. So the dotted line is basically a one-step growth curve. So you can see the little eclipse period here, and then you get production of infectious viruses. It ends about 6 hours after infection. The solid line is the radioactivity incorporated into RNA. And so here he's labeled the y-axis RNA polymerase activity because he figured out eventually what it was. But he saw for the first time that in a virus-infected cell, an RNA virus-infected cell, you got RNA made during infection. And this RNA was resistant to a drug that was known to inhibit cellular mRNA synthesis. So this polymerization was not being done by a cellular enzyme. So this was the first evidence that viruses encode an RNA polymerase. Later, the same guy, Baltimore, discovered an RNA polymerase in the particles of negative strand viruses. So here we have vesicular stomatitis virus, one of the viruses we've talked about. This is a negative strand virus. So Baltimore reasoned, well, plus strand viruses, the RNA is messaged, that gets into a cell, you get a polymerase made. But negative strand viruses can't, their genome can't be translated, so they must have the polymerase in the particle. So that was his reasoning. So he looked in the particle, he purified the particles, he cracked them open slightly with a little detergent to let the radioactive triphosphates in and incubated them, and lo and behold, they had a polymerase in them as well. Subsequently, you can, if you identify a new virus and sequence its genome, you could look at the sequence of the proteins, and if you find certain motifs like GlyAsp-Asp, these tell you that that's probably an RNA polymerase. So there are certain amino acid motifs in RNA-dependent RNA polymerases that can help you. And then, of course, you can simply put the gene encoding what you think is the polymerase into a vector and produce it in cells and show that there's RNA polymerase made. Today, actually, many people study RNA polymerases of different viruses. They're good targets for antivirals because no such enzyme exists in the cell. There's no RNA-dependent RNA polymerase. And so the related enzyme of HIV, reverse transcriptase, which copies RNA into DNA, there are tons of inhibitors for that enzyme because it doesn't exist in the cell. So we have lots of crystal structures of these enzymes because that helps you design antivirals. We'll talk about that in a separate lecture. So among all the RNA viruses, okay, so all these viruses have to encode an RNA polymerase, as we said before, because the cell cannot copy their genomes. The RNA looks slightly different depending on the polarity of the genome. So negative strand RNA genomes in the particle are always coated with protein. So vesicular stomatitis virus, Ebola virus, they not only have a polymerase, which is shown in the VSV illustration as the little sphere, but the RNA is also coated with protein. You know that already because I told you these viruses are built with helical symmetry, right? A single protein wrapping around the RNA. Plus strand genomes are typically naked in the particle. They don't have proteins associated with them because you don't need to. The RNA gets in the cell and it's immediately translated. There's no need for any proteins, no polymerase or anything to be associated with it. There are two exceptions that you need to know, retroviruses and coronaviruses. So here's poliovirus on the left. Its RNA is naked. He's a flavivirus. Its RNA is naked. But the retrovirus, the RNA is complexed with a protein as if it were a negative strand virus. Now these viruses have to be reverse transcribed to DNA when the RNA gets into the cell. Remember, although they have plus stranded RNA, it's not translated. It's reverse transcribed. So probably the protein is present to facilitate that process. So that explains that exception. But coronaviruses here on the right, their plus strand genome is also coated with protein. And we don't really understand why this is. Coronaviruses are like SARS and MERS coronavirus, the one that's wreaking havoc in the Middle East at the moment. We don't know why there's protein on it. The one possibility is that these genomes are so long, maybe they need to be protected by an RNA shield. So they're the longest RNA genomes, these coronaviruses, 27 to 30 kilobases, far longer than any other RNA virus. So maybe it's to help stabilize them, but we really don't know. Finally, the double-stranded RNA genomes are not complexed with protein in the particle. They're present as just naked double-stranded DNA, although these double-stranded viruses do have a polymerase in the particle, right? Because double-stranded RNA cannot be translated. You have to make a message from it. The polymerase seems to be attached to the particle shell, the inner side of the particle cell. In fact, there seems to be a molecule of RNA polymerase at each of the five-fold axes of symmetry. We'll talk about this in a bit. So it's not attached to the double-stranded RNA in itself. So these are some rules that you can remember, but as always, there are exceptions. Just to remind you, here's how the protein coats the genome of vesicular stomatitis virus. Remember, these are bullet-shaped particles. The RNA genome, the negative strand RNA genome, is bound to a single protein called nucleocapsid protein, and those proteins interact with each other and with the RNA to give the helical symmetry. The way this works is shown at the top. The RNA is shown in yellow, and then these individual molecules of nucleocapsid protein are showing shown binding the RNA. There's a groove in the protein that binds the RNA, and then the proteins interact with each other. So lots of interactions, a very stable nucleocapsid. Are these covalent interactions, by the way? They are not, because they have to come apart at some point when RNA polymerization is happening. On the right is influenza virus, another negative stranded RNA virus, and its genome, again, is complexed with proteins. This genome is in 8 pieces. There's also a polymerase at the end of each RNA segment, as you can see in the diagram. At the top is the structure of the RNA protein complex determined by a combination of X-ray crystallography and cryo-EM. And this is just a piece of RNA that's only long enough to bind 1, 2, 3, 4, 5, 6, 7 end protein subunits. But it shows you how these are organized. And at the end here, this large lump is the RNA polymerase. So it's at each segment ready to copy the RNA. So that's how nucleocapsids are built in general. In addition to having protein on them, or not, of course, RNAs are structured. We talked about this very briefly before. They can have a secondary structure which is based on base pairing within the same molecule of RNA. So here, for example, is a stem loop, where you have base pairing among adjacent sequences in the RNA, so it forms that kind of a structure. These can be quite complicated. They can be extended. You can have interior bulges. You can have branches, and so forth. You can even have something that's called a pseudoknot. Here you have a stem loop, and residues in the loop are base pairing with downstream sequences. So you get this sort of structure. And it looks like a knot, but it isn't really, so that's why it's called a pseudoknot. That's the three-dimensional structure of it. All of these have importance in the viral replication cycles. They tend to be sites for protein interactions during replication or translation, and they may also interact with each other. These loops can also undergo long distance interactions, so they're very important. A couple of general rules about RNA synthesis. First, the genome has to be copied end to end with no loss of sequence. That's obvious, right? If you lost sequence every time you copied the genome, you wouldn't have any infectivity at some point. It's sort of like the chromosome end problem. You know, if you didn't have telomeres, you would lose the ends of chromosomes as DNA replicated. So you have to copy, if this is an RNA genome, you have to copy it end to end. And you also have to make mRNAs that can be translated by the cellular ribosome. Right? It has to be translatable, and there's some requirements for that as well. So these are two fundamental issues with respect to RNA synthesis. There are some universal rules as well. Synthesis begins and ends at very specific sites on the template. It could be the end, it could be an internal site, but it's always specific. The polymerase, RNA-dependent RNA polymerase, may initiate what we call de novo. That means without a primer, or it may require a primer. Okay, so it depends on the virus enzyme. There's no way you can remember it really, but we're going to talk about some examples of de novo versus priming. Sometimes other proteins are needed to carry out RNA synthesis. These can be other viral proteins. These can be cellular proteins as well. And then this one here, you should know, RNA is synthesized by incorporation of NTPs, and the new strand is made in a 5' to 3' direction. The template, of course, is copied from the 3' end to the 5' end. It's the same for DNA. There are also examples of non-templated RNA synthesis. So in all of these rules here, we're talking about a template. In other words, the polymerase is copying a template and making a copy strand. But sometimes there is RNA synthesis that is not based on the sequence in the template. We call that non-templated. And at the end today, we'll talk about an example of that and what it means. So here are some examples of priming, just to get you thinking about how that works. We have it divided into de novo initiation. De novo simply means new, without a primer. You don't need a primer, and primer dependent. So in de novo initiation, the simplest case, you have a viral RNA. Here's 3' end. The polymerase can simply recognize that last base and find the NTP that's complementary, and then add the second one and so forth. So it doesn't need a primer. It can just say, yeah, that's a U, and we'll put an A there, and so forth. So that's beginning right at the end. Sometimes initiation begins internally. On some viral RNAs like this one, It's starting at the sixth base from the sixth base from the 3' end. It extends a little, and then it actually slips back, so it doesn't lose sequence at the 3' end. This is just an unusual mechanism. It's nothing special. So those are two de novo initiations. And here we have two different kinds of primer-dependent initiation. Here, in one case, the primer is actually linked to a protein. And in this case, terminal protein, it could be, the coronaviruses, for example, use a protein as a primer. Then the first nucleotide is added onto it, and you'll see a little bit more about that today. Or the primer can be a capped piece of RNA. Caps are structures on the 5' end of messenger RNAs, and some viruses actually use those as primers. We'll talk about these in some detail. Now, the way that nucleoside triphosphates are incorporated into the growing strand of RNA is very much the same as for DNA. It's catalyzed by what we call a two-metal mechanism of catalysis. So here, let's start in the upper left. Here's a strand of this. This is actually the DNA model, because you can see there are T's here instead of U, but the same applies for RNA. So on the upper left, we have a template strand, and we have a primer initiating synthesis of a complementary strand. And triphosphates are brought in and incorporated depending on the sequence of the template, of course. And here we're looking at a very small piece of the extended reaction. Here's the template strand on the right from the 3' to the 5' end. And here is the newly synthesized strand from the 5' end down. And we've put in a complementary C and an A. And now the next base, the T, is being incorporated. It's added, of course, as a triphosphate. Here are the 3 phosphates shown here, the alpha, the beta, and the gamma phosphates. And the way these are added is by a two-metal mechanism. And here are two metals that are essential for catalysis. They are held in place by two aspartate residues. You can see them here, held in place by the polymerase. These aspartates are in the polymerase. They're in DNA polymerases. You'll see they're also in RNA polymerases. And the magnesium metals help to maintain, help to carry out the reaction. There are a series of nucleophilic attacks which eventually eliminate the beta and the gamma phosphates and link this phosphate to the preceding base, so that now you have two bases with one phosphate in between them. So we'll see where these metals are located in a moment, which is a universal rule about RNA-directed RNA synthesis. 1, the polymerase may initiate de novo or require a primer. 2, synthesis initiates randomly on the template. 3, RNA is synthesized in the 3' to 5' direction. And 4, synthesis is always template directed. Okay, I don't know why there are more answers than people in the room. That's weird. All right, most of you got, 84% of you got A. The polymerase may initiate de novo or require a primer. That's certainly correct. The rest are not. The synthesis is not random on the template. There are always specific sites of initiation. 3 to 5' direction. The new RNA is synthesized in a 5 to 3' direction. The template is read in a 3 to 5' direction. It's just, you just have to remember that. There's no easy way. And 5, synthesis is always template directed. There are some examples of non-templated RNA synthesis that I mentioned that we'll talk about in detail later. Now, there are 4 different kinds of nucleic acid polymerases. The one we're talking about today, RNA-dependent RNA polymerase. And this is just kind of a linear map of the sequence of the protein. RNA-dependent DNA polymerase, that's reverse transcriptase. We're copying RNA into DNA. DNA-dependent, so the dependent part is the template, of course. DNA-dependent DNA polymerase, so it makes DNA from DNA. And finally, DNA-dependent RNA polymerase. That is the enzyme that makes messenger RNAs in a eukaryotic cell, for example. 4 different classes. We've identified all 4. We've cloned them and sequenced them. We have crystal structures of all 4 of them. They have some conserved motifs, which are shown in colors here, A, B, C, D, and E in some of them. The rest of the protein is not terribly similar, but they all look pretty much the same in their 3-dimensional structures, so they may have evolved from a common ancestor many, many years ago. In the C motif in yellow is that amino acid motif, which if you see can help you identify that you're looking at an RNA polymerase. And so, for example, in plus-stranded RNA virus polymerases, it's a gly-asp-asp, GDD, okay? That is, if you see that in a protein, you can pretty much bet it's an RNA polymerase. And for other viruses, negative-stranded, segmented viruses, it's an asp-asp or a gly-asp-asp in others. You don't need to remember these. You just need to know that in this C area, there is a sequence motif. It turns out that the aspartates are the aspartates that coordinate the magnesium, and that's why they're so conserved. So here is the three-dimensional structure of the RNA-dependent RNA polymerase of poliovirus, which was the first one of this class to be determined. And this is characterized, like all the other polymerases, as a right hand. So they all supposedly look like a right hand. They have fingers, thumb, and palm domain. The palm domain contains the active site. So these conserved sequences or motifs on the 4 different kinds of polymerases, they're colored and you can see where they're located on the structure. So for example, A motif in red is right there, but the one I want to show you is the C motif, which has that gly-asp motif. It's shown in yellow right here, and here are the two aspartate residues shown as a stick figure, and that's where the magnesium ion is going to be coordinated. So this is the active site of the polymerase. So you can see that the highly conserved motifs, while they're far away in the linear sequence, they're actually all together in the active site. So they constitute the active site. So nucleic acid polymerization, the core part of the enzyme that is catalytically active, is very similar among the 4 different classes of enzyme. And the other parts of the molecule diverge, and they have various functions. They have to interact with different proteins and so forth. So this is a space-filling model of the poliovirus RNA-dependent RNA polymerase. With just a little bit of the front cut away so that you can see what's going on here. And what's happening here is that the template is in cyan, and that's coming in through a tunnel in the bottom of the molecule. And it's passing over the active site. It's a little bit obscured here. But this purple coloring, I guess that would be magenta, is where the 2 aspartate residues are. In fact, you can see one of them in the red sphere. So the 2 aspartates are there. Remember, they're coordinating the magnesium. And that's where the triphosphates are brought in and added to the growing chain. This is the catalytic center. The product chain is shown in gold here, and it is being elongated. There's the end of it right there. Sorry, the product is, yeah, it's in gold, and it will come out the top of the molecule. So this opening here is a natural opening in the top of the molecule, and the product comes out of the top. Okay, so that's more or less how the topography is. Now, the structure is very useful for many things. You can design inhibitors, as I told you, but it also tells you how the reaction works. So these RNA-dependent RNA polymerases do not accept deoxy-NTPs. That's what's used for DNA synthesis, right? They only accept NTPs. And when you look at the structure, you can see why. So this is the catalytic site of the enzyme. And here is a UTP bound in the catalytic site, which is about to be added to the growing chain. All right, so it's carrying out, or it's engaged in hydrogen bonding with amino acid side chains of the polymerase. You can see at the top here and also at the bottom. Here, by the way, are the 2 aspartate residues that coordinate the magnesium ions. But the one I want to show you is this D238, which is making a hydrogen bond with the 2' hydroxyl of the sugar of the UTP. So here's the chemical structure of it. Sorry, it's backwards. I had to flip it around so it would be in the same orientation as the actual structure here. The triphosphate, the sugar, and the base. So here's the 2' hydroxyl base pairing with, hydrogen bonding with D238. If this were TTP, it would not have an oxygen here. Would just have a hydrogen, would not be able to hydrogen bond. So that's why the DNTPs are not accepted. If you change that residue in some of the polymerases, you can get the enzyme to recognize DNTPs. So it's a really interesting explanation for the specificity. Now let's talk about the overview of how RNA synthesis occurs. That's all the molecular detail for the polymerase that we're going to get into. I want to go through plus, minus, and double-strand RNA viruses and tell you how they make RNA in the cell. So we're going to start with plus-stranded RNA viruses, such as the picornaviruses, poliovirus, and flaviviruses, both of which we've seen in structure and in cell entry. These have plus-strand RNA genomes, and they're copied in a very straightforward manner. The plus strand is the message and the genome. You make a negative strand intermediate, and you copy that again to get more plus strands. Really straightforward. That negative strand, its role in the world is simply to be a template to make more plus strands. It doesn't do anything else as far as we can tell during replication. So the picornaviruses get into cells. They put their RNA in the cytoplasm by mechanisms we talked about last time. And because it's a plus RNA, it can be immediately translated. It's engaged by ribosomes. You make a protein. You actually make, for these viruses, a long protein called a polyprotein, which is then cleaved by viral proteases to get all the individual proteins that you need. Among those proteins are the RNA polymerase, which then have to go on and copy the RNA and make more copies of it. So they take the plus strand RNA that comes in, they make a negative strand copy. It's shown here associated with the plus RNA as a double strand. And then the negative strand is copied again to make more plus strands, which eventually will be either translated. Again, some of these RNAs that are made could go back into the translation pool, or they can be packaged into new virus particles at the end of the cycle. This is a relatively straightforward replication cycle. One unique aspect here is that RNA synthesis is shown here occurring on double membrane vesicles. For all the plus-stranded RNA viruses, at least, RNA synthesis seems to occur on the surface of vesicles. These are, of course, spheres in the cell. They're derived from cellular membranes because the virus can't produce them on their own. And it seems that it's a good place for very efficient RNA synthesis, because if you inhibit vesicle formation, you don't get RNA synthesis. So here's a schematic of the picornavirus RNA genome. Again, it's a plus-stranded RNA. It is a messenger RNA. It can be translated directly in the cell to make a very long polyprotein. And then within this protein, there are two proteases, one here called 2A and one here called 3C. And these actually chop up the long protein into smaller proteins like capsid proteins and everything else you need to replicate. Here's the RNA polymerase. It's called 3D Pol. It's all the way at the end of the genome. Now you'll see there's some common features of this RNA. There's a 5' and a 3' untranslated region. There's a poly A tail. But at the 5' end, there's a protein linked to the RNA. This is very unusual. And you will see that gets there during priming, because the primer for RNA synthesis of these viruses is a protein-linked primer. Now the genome, in another way, looks like this. The genome actually has lots of secondary structure, and 3 of them are shown here. At the 5' end, there's this stem loop structure, which has been called the clover leaf. In the middle, there's a stem loop called the cis-acting RNA element, and at the 3' end, there is a pseudoknot. And these are thought to contribute to the specificity of the reaction. The RNA polymerase made in infected cells does not copy any messenger RNA in the cell. Remember, messenger RNAs are plus-stranded, they're polyadenylated, but they're not copied at all by the viral polymerase. The polymerase only copies viral RNAs, and it's probably because of these structures that make the copying reactions very specific. Let's talk a little bit about this clover leaf at the 5'. Here's the actual sequence of the clover leaf. And the very first base of the viral genome, which is a U, is linked to the protein by the hydroxyl. It's actually a tyrosine in the protein that is linked to the first phosphate. So this is the small protein linked to the 5' end of the genome through this kind of linkage. And then the rest of the genome follows. The way the RNA of these viruses replicates is very interesting. It occurs on membranes. That's schematized on the upper left here. And one of the viral proteins is a membrane-bound protein that has affinity for the replication complex. And in particular, there is a cellular protein called PCBP, which binds to the clover leaf at the 5' end of the viral RNA. And that seems to anchor the whole RNA to the vesicles so it can be replicated there. The RNA, when it's copied, actually forms a circular configuration. And that is because poly A binding protein, which is a cellular protein, binds to the poly A at the 3' end of the viral RNA. It in turn binds to the viral RNA polymerase, shown as 3CD-Pro, which is binding to the clover leaf, and that circularizes the RNA. So what happens is, in this circular form, which is essential for RNA synthesis, the RNA polymerase adds 2 U residues onto VPG, which is the protein linked to the 5' end of the genome. So VPG, of course, is made in the infected cell. The polymerase takes it, uses the poly-A template to add 2 U's, and then that serves as a primer to elongate the new strand. So VPG PU is the primer for the viral RNA polymerase. It's a protein with 2 U residues that are complementary to the poly-A strand. So the polymerase is now shown here using this primer to elongate on the template, and of course eventually will make a full-length negative stranded copy of this plus-stranded RNA. So it's an unusual protein primed reaction that also requires a circular template. Why, we don't know. But if you block the circularization, as I said, you don't get RNA synthesis. When you make the other strand, when you have to take the minus strand and make a plus strand from it, the VPG is uridylated on this CRE, this cis-acting RNA element, which is a stem loop in the middle of the genome. So normally, for the copying the plus strand, the VPG is uridylated, that is 2 U's are added to it using the poly-A as a template. For some reason, to do the other strand, well, there's no more poly-A, of course, at the 3' end, so perhaps that's one of the reasons. This CRE element in the middle of the genome serves to uridylate VPG. So the CRE gets covered by polymerase molecules. That's what these blue guys are here. VPG comes in. This stem loop in the loop region is A-rich. So the polymerase simply uses the A template in the loop to add 2 U's onto VPG, and then that serves as a primer for plus-strand RNA synthesis. So there are 2 different modes of uridylating this primer. Now, as I said before, RNA synthesis in plus-strand virus-infected cells always occurs on vesicles. And these are 3 different images of the kinds of vesicles that you see here. On the top is poliovirus infected cell. On the left, these are high magnification EMs. On the left is an uninfected HeLa cell. And there's lots of endoplasmic reticulum and Golgi in these cells. But after a few hours of polio infection, all the ER and Golgi is gone. It's replaced by these double membrane vesicles. And those are the sites of poliovirus replication. You can actually see new particles have already been formed in this infected cell. It's late enough in replication. Here on the bottom left is a similar vesicle in a flavivirus infected cell. Here's an electron micrograph. You can see these multiple vesicles. This is where RNA synthesis occurs. And production of new particles is very tightly linked to that. You can see a new particle being formed here. On the right is a 3-dimensional reconstruction of this EM using graphics. And this is a coronavirus infected cell where, again, you get lots of vesicles forming on which RNA synthesis occurs. So RNA synthesis on vesicles is essential. And as I said, we think it's because you can concentrate all the components very effectively on a surface rather than having them floating around randomly in a cell and having to depend on bumping into one another for the reaction to occur, which is a part of the poliovirus replication strategy. One, the production of subgenomic mRNAs. Subgenomic means shorter than the genome RNA. De novo, without primer initiation of RNA synthesis. Circularization of template for initiation of RNA synthesis, or maybe all of these could be right. Okay, so the correct answer is C, which most of you got. Circularization of template for initiation of RNA synthesis. Subgenomic mRNAs are not produced in picornavirus infected cells. As I said from that first slide, it's a very straightforward strategy. You have the plus-stranded genome, you make a full-length minus strand, then you copy it again. So there's no subgenomic mRNA. De novo initiation means without a primer, and of course I showed you that the primer is VPG with 2 U's on it, and so obviously it can't be all of the above. Okay, so those are picornaviruses. Let's look at another example of a plus-stranded RNA genome that does it a little bit differently. These are the alpha viruses, and we've talked a little bit about, as a model, Semliki forest virus, and chikungunya virus is another example of one of these. So these all have plus-stranded RNA genomes in the particle, so they are not covered with protein. They are copied to form a negative strand complement in the infected cell. And that, interestingly, well, that of course is done, that replication is done by translation of part of the plus-stranded RNA, as you will see. And then the negative strand serves as a template not only for a new plus strand, but also for mRNAs. So let's take a look at how this happens. Here's an alpha virus infected cell. These enter by receptor-mediated endocytosis. The RNA ends up in the cytoplasm. Plus-stranded can be directly translated. So here on the left, we're seeing the RNA being translated, and the one protein that is made initially is the RNA polymerase. It's a 4-subunit enzyme shown here. The RNA, so the genome is translated, so it's a plus-stranded RNA. That RNA polymerase that is made then copies the plus strand into a negative strand complement on vesicles, as I said. That can serve as a template for making new full-length plus strands, which eventually are going to be used to make new virus particles. But these negative strands have an additional role, and that is they're templates for the synthesis of a subgenomic mRNA, and that's shown here. Subgenomic simply means that it's shorter than the genome. So poliovirus does not make any subgenomic mRNAs. Its mRNA is the genome, that's it. But these viruses make a smaller RNA, and that serves to produce the structural proteins, the capsid protein and the glycoproteins. So let's see how that works. Here's the viral RNA plus-stranded in green. It's capped at the 5' end, poly A at the 3' end, untranslated regions. So this one is not a protein-primed reaction. As this RNA gets in the cell, the first half or so is translated to make the RNA polymerase and the other proteins that are needed to make RNA. That RNA polymerase then copies the plus strand. You get a full-length minus strand that is shown in olive right below it. And then the polymerase again copies that minus strand, but at this promoter, it's shown in red here, to make this subgenomic mRNA. All right, so that gives rise to the structural proteins of the particle. Now you may ask, why don't you just make a polyprotein? Well, you know, why questions you can't really ask. So you would have to say, what's the function of not making a polyprotein? And I would say, well, there's a stop codon right here, and that stops translation. And so you have to have another way to access this coding region, and you do that by making a subgenomic mRNA. We don't have a really good answer for why that stop codon arose and so forth, but this is just another strategy that happens to work. So that's a subgenomic RNA. You can see it's shorter than the genome RNA. Okay? So those are two examples of plus-stranded RNA replication strategies. Let's go to minus-strand viruses. We're going to look at two different kinds. We're going to look at minus-strand viruses that have one genome, one piece of RNA, and it's shown in the middle here, the olive color, or some that are segmented. So remember, the minus-strand viruses have either unimolecular or segmented RNA genomes, but the replication strategy is similar. You have a minus-strand genome RNA for both. mRNAs are made to get proteins produced in the infected cell in both cases. And then to replicate that genome, you go through a plus-stranded intermediate, just the opposite of what you would get for a plus-stranded RNA virus. So these full-length plus strands exist solely to be templates for the synthesis of more minus strands. So let's see how that works for two different examples. We'll start with a unimolecular RNA, vesicular stomatitis virus, rabies virus. These are the bullet-shaped viruses. They have a polymerase in the particle, right, because it's a negative RNA. And as soon as that RNA gets in the cell, it has an associated polymerase that makes messenger RNAs, because you have to make some proteins to replicate. Now, the strategy for this virus is to make multiple subgenomic mRNAs. You can see 1, 2, 3, 4, 5 subgenomic mRNAs. All right? So you start with one long negative strand RNA, you make 5 subgenomic mRNAs, and each of those gives rise to a protein, proteins to make the polymerase or to make more virus particles. But you obviously cannot make new viruses from subgenomic mRNAs. They're not long enough to make the genome. So you have to also make a plus strand copy of the negative strand genome, which is shown down here. That's copied again to make the minus strands, and those eventually can be put into new virus particles. So it's just another way to express genetic information. So far we've seen a couple, and one is a polyprotein to make lots of proteins from one mRNA. A subgenomic mRNA is another strategy, and here you're making multiple subgenomic RNAs because you have to figure out how to get these proteins made from a single RNA that's in the virus particle. So here's a schematic of the VSV RNA. Again, negative strand shown in olive color, and then the virion, of course, that's all wrapped up with a nucleocapsid protein to make that lovely helical structure, which you can see in both images. That RNA is complexed with a polymerase, shown here made up of 2 proteins. And as soon as that RNA gets in the cell, the polymerase is at the 3' end, it's ready to copy the RNA, and it makes subgenomic RNA, as you can see here. 1, 2, 3, 4, 5. And these encode, again, the polymerase, which is the L protein, and then lots of structural proteins. Let's look at this in a little bit of detail to see how it works, because we understand this process quite nicely. Here's a schematic of mRNA and genome synthesis for VSV. So the genome that comes in with the virus is shown, the second line. It's the olive-colored RNA, and that's labeled negative strand genome RNA. It has the polymerase, which is a complex of a variety of proteins, the polymerase itself and P proteins. It's already stuck on the 3' end of the genome as it comes in the cell. So as soon as that is released into the cell, then there are triphosphates around, NTPs. There are no NTPs in the particles, so no RNA synthesis occurs. So as soon as it's released, it finds NTPs, it can start making messenger RNAs. So again, the 5 are on the top. You can see they're each made, and each of those encodes a separate protein. Now, I told you before that you have to make a full-length plus strand copy of that negative strand RNA in order to replicate the genome. The full-length plus strand copy is shown here in green. So at some point in the replication cycle, there has to be a switch from mRNA synthesis to making genomes so that you can make more viruses. And typically this happens when you've accumulated enough structural proteins to make particles. You have to have the glycoproteins that are going to be embedded in the particle, the internal structural proteins, and so forth. So as soon as the virus comes in, there's no point in making genomes. So the key here is the switch from mRNA synthesis to genome synthesis. And the way the switch happens, or a major determinant of the switch, is one of the viral proteins called the N protein. And as its concentration rises, the switch to full-length plus strand synthesis occurs. So remember, initially the polymerase is making subgenomic plus strands. At some point, it makes a full-length plus strand that covers the entire genome. It does that when the concentrations of the N protein reach a certain concentration, and that begins to coat the plus strands that are made. So on the top, when there's no N protein or very little N protein around, it can't coat the new mRNAs, and so the polymerase terminates and makes separate mRNAs. But as soon as N protein, which is this little sphere with the blue interior, as soon as it's made, as soon as the enzyme begins to make a new plus-stranded copy off the negative strand template, the N proteins bind to it, and they cause anti-termination of the polymerase. So the polymerase no longer stops after each mRNA, and you get a full-length plus strand. And that can be copied in turn to make the minus strand, which you need to make new genomes. So again, the switch from mRNA synthesis to genome, full-length genome synthesis is mediated by the N protein, which serves to anti-terminate at the ends of the mRNAs. So again, individual mRNAs initially, and a long plus strand later on when the N protein concentration gets high. Let's take a little bit more look at this in detail. At the top is your genome again, as it comes in the virus. Here's the 3' end, and the polymerase is bound to it. Between each gene is what is called an intergenic region. It's a sequence that tells the polymerase to stop. So the polymerase initially starts to copy the 3' end of this first gene, the N gene. It makes an N mRNA. And then it terminates at the intergenic sequence. So now we have a unique mRNA, and then it begins to make the pMRNA. And at the end of that sequence, it reaches the intergenic sequence, and it will terminate. So that's how it goes down the genome, making an mRNA, starting and stopping, and then simply moving down. And it's that process that's antagonized by the N protein. The N protein says don't stop at the intergenic sequence, and that's why you make a long plus strand later in infection. Now, polyadenylation was also specified within this intergenic region. So these are typical mRNAs. They have a cap at their 5' end, which is not shown here. They're polyadenylated. And the polyadenylation is specified by the intergenic region. Here is the sequence of one of the intergenic regions here at the top. So again, here is our 3' end of our negative strand genome RNA. The polymerase is in the process of making an mRNA, and it's reached the intergenic region. There is a series of U residues. You can see them here, 1, 2, 3, 4, 5, 6, 7. And for some reason, when the polymerase hits that, it begins to slip, and it doesn't move forward. It just sits on the U's and makes A's over and over again until you get about 200 or so A's, and that's a poly A tail, and then the polymerase stops. It's fed up. I said, I'm not making any more A's. And the polymerase stops, and that mRNA is released. So that's how it gets polyadenylated. And then the polymerase moves on and initiates at the next gene. You can see the next mRNA is being initiated here. Here's the cap and the actual coding sequence of the mRNA. So polyadenylation, termination and polyadenylation is specified by the intergenic region by slippage on that U sequence. That's going to be similar to what we see for influenza viruses in a moment. Now, influenza viruses are more complicated because there are 8 copies of the genome RNA. So everything is multiplied and makes for kind of confusing figures such as this. The other complication is that this virus, as opposed to all the others we've talked about so far today, has to get into the nucleus to replicate its genome. And that has to do with the primers that are made for replication. I'll tell you about that in a moment. So these viruses enter by endocytosis. We talked a lot about this process last time. The genome enters the nucleus. So 8 pieces of RNA, negative strand RNA complex with proteins, enter the nucleus. Each one of those has a polymerase associated with it. So a messenger RNA is made for each one. And then the resulting proteins are made, and they of course have to be made out in the cytoplasm. So each mRNA that's produced has to be exported to the cytoplasm. Proteins are made in the cytoplasm, and some of those proteins have to go back in the nucleus to form more helical ribonucleoproteins. But those mRNAs that are made, and there are 8 of them, those are not complete copies of the minus strand. So at some point you have to make full-length plus strands, plus-stranded copies of these genomes, which are shown here, and those can be used to make more negative strands. All right, so all of this is happening in the nucleus. Here's the schematic of the genome. Here, the 8 negative-stranded RNAs, they're again wrapped up in protein and have a polymerase on each one. So they're helical ribonucleoproteins. And on the left is a 3-dimensional model, which kind of shows you nicely what this particle looks like. It's cut open, and you can see the coiled up ribonucleoproteins, or the nucleocapsids, inside of the particle. So each of these negative strand RNAs that are in the particle, they get out into the nucleus. They're copied into an mRNA by the viral polymerase, which is associated with each segment. And for the most part, each mRNA encodes one protein. There are a couple of exceptions where alternative reading frames or splicing gives rise to 2 proteins per segment, like segments 7 and 8. But pretty much it's one segment, one protein. So this is another strategy for making multiple proteins from a viral genome. Some viruses have one RNA, and we've talked about the strategies to translate that. And this one, the approach is to make the RNAs in pieces. So you correspond one protein, more or less one segment of RNA. Let's look at how this process happens for each segment. It's very similar to VSV, but there's some interesting twists. Here's the negative strand RNA genome. Of course, it comes in the particle with a polymerase attached to the 3' end. This is a primer-dependent RNA polymerase. The VSV polymerase was not primer-dependent, doesn't need a primer. The flu polymerase needs one. And the primer that it utilizes is a piece of RNA with a cap on it that's derived from host cell messenger RNAs. So this is 12 to 14 nucleotides long. It's got a cap on the 5' end. And the virus polymerase actually cleaves this from cellular messages. And that's why the replication in part has to happen in the nucleus, because it picks up these freshly made mRNAs in the cell nucleus. So this acts as a primer, and you have mRNA synthesis at the top. So each segment is transcribed into an mRNA. Because of the primer usage, the 5' end of every viral message is different. It's not viral, it's cellular, and it comes from any cellular messenger RNA. So it's a mixture of sequences, but it's a non-coding region, so it doesn't matter. The 3' end falls short of the genome RNA. It's about 20 nucleotides short. So the virus can make all of these mRNAs, one per segment, make plenty of proteins, but at some point, if you want to make new viruses, new virus particles, you have to copy the negative strand completely. These mRNAs can't be copied again to make negative strands because the 3' end is too short. So as you might predict, these negative strands are copied to full-length plus strands. So again, just like for VSV, you have to have a switch from mRNA synthesis to what we call replication, production of full-length plus strands, and from those full-length minus strands. And just like VSV, the protein that coats the RNA controls the switch. In this case, it's called nucleoprotein, but the principle is the same. As these mRNAs are translated, the levels of these proteins rise, and then at some point, the protein begins to coat the newly synthesized RNA, and it causes anti-termination at the 3' end, so now you have a full-length plus strand. These full-length genome copies are not primed with a cap. They're unprimed, so in certain conditions, the polymerase can work without a primer, that is, when it's making full-length plus strands. And from those, you get full-length negative strands, which are used to build new virus particles. So let's take a look at this cap. It's quite interesting that the virus uses caps to prime its mRNA. And this is a schematic of that reaction. So again, it takes any cellular mRNA. It doesn't matter what it is. Here's one particular cellular RNA. There's a cap structure. It's simply a G residue linked in a 5' to 5' direction rather than a 5' to 3'. And we'll talk about the function of those later. And then you go into the rest of the messenger RNA there. The viral polymerase, which is a multi-subunit enzyme, has in it an endonuclease, which can cleave host messenger RNAs to give rise to the primers. So here's one cleavage site, and then you get a primer, a cap with 13 bases in this case, and then that is utilized by the polymerase to prime on negative strand RNA to make messenger RNAs. So all the messenger RNAs, again, have a little bit of cellular message at their 5' end. And again, this doesn't happen for full-length plus strand synthesis. That is primer independent. So this has been lovingly called cap snatching by the person who discovered it about 40 years ago. And now it's been found in other viruses as well. Polyadenylation also occurs by a stuttering mechanism. Now, when you think of a RNA being copied by a polymerase. So here my arm is the RNA, polymerase is moving down. This is the classical model, is that the polymerase is moving. But we think for some, in particular flu, the polymerase is stationary and the RNA is being pulled through it. So here's the polymerase of flu on the top. There are 3 subunits, and one of those subunits is the endonuclease that cleaves the caps, another one is catalytic. And here is the viral minus strand RNA being copied into a message. So the minus strand RNA is bound to the polymerase at its 5' end, and the 3' end is passing by a catalytic site, which is this red sequence right here. So the 3' end is being pulled through, and as it's pulled through, a messenger RNA is being made. So it starts at the 3' end, of course, and here we're about halfway down through the vRNA, the negative strand RNA. We're pulling the negative strand through, and if the 5' end is anchored, which is the case, eventually you can't pull it through anymore. And it so happens that when it's in this configuration, where it can no longer be threaded through the polymerase, there's a stretch of U on the minus strand template. And that stretch of U serves as a template for the synthesis of poly A. The enzyme begins to stutter. It can't move anymore because the template is locked, and you make a poly-A sequence. All right, this one we're saying, how are influenza virus and VSV RNA synthesis similar? One, the switch from mRNA to genome RNA synthesis is controlled by an RNA binding protein. Two, polyadenylation occurs at a short stretch of U residues. Three, viral mRNAs are shorter than minus genome RNA, or all of the above. So 70% of you got D, which is all of the above, and that's the correct answer because every one of these is in common. The switch is controlled by an N, or nucleoprotein, an RNA binding protein that anti-terminates. Polyadenylation occurs at a short stretch of U's, whether it's in the intergenic region for VSV or where the RNA is being threaded through the polymerase for flu. And the viral RNAs are shorter than the genome RNA. That's why we need a switch between mRNA synthesis and genome RNA synthesis, because the mRNAs are not long enough to be substrates for replication. The last virus I want to talk about are the double-stranded RNA viruses, reovirus, rotavirus. And this is unique because the double strand, although it contains a plus and a minus, the plus can't be translated directly. So what has to happen, a polymerase has to be in the particle. In the particle, it will copy the negative strand in the duplex and make a messenger RNA. That will be translated into protein. And then to make more genomes, some of those messenger RNAs are simply copied again to make duplexes. So it's unusual because of the double strand. And here's how it works. These viruses get into cells by endocytosis. And last time we talked about these viruses as actually wanting to remain in the endocytic pathway even after the lysosomes are fusing. Most viruses want to get out of the pathway because lysosomes are degradative, but these viruses want to be degraded. Of course, they don't want to be, but that's the way it happens, right? So here, the virus outer shell is being removed by the degradative enzymes in the lysosome. So we start with a double-shelled particle with an outer and inner icosahedral shell, and we end up with just the core. The outer shell is removed by lysosomal enzymes. The core is hydrophobic, can poke out into the cytoplasm. So now we have this core, which has all the double-stranded RNAs. Remember, there are multiple segments. And the enzyme is in the polymerase. Sorry, the enzyme is in the particle. And it starts to make mRNAs, and they come out through these turrets at each five-fold axis of symmetry. So the mRNAs come out into the cytoplasm. The genome never gets out of the particle, right? It stays inside the particle, and the mRNAs come out. Those are translated to proteins. You then can build new particles using the proteins, and you put single-stranded mRNAs into those particles. And among the proteins in the particle is a new polymerase, so that can make the RNA double-stranded, and then eventually you can make another shell on the outside and get new virus particles. So the neat thing here is that the mRNAs remain in the core. Sorry, the double-stranded RNAs remain in the core. The mRNAs are extruded. Those are used to build new particles with mRNAs in them, and a shell, of course. They're made double-stranded, and then you make new virus particles. So the mRNAs come out of the particle at these five-fold axes of symmetry. We actually think that there is a polymerase bound to each five-fold axis of symmetry, and we think there may be an RNA then associated with each polymerase. Do you remember how many five-fold axes of symmetry there are in an icosahedral particle? 12. No virus with more than 12 segments has ever been found. And we think the limitation is the number of five-fold axes of symmetry. You see, I just didn't want you to memorize that number. There was a reason. Okay? Just memorization for its own sake is no good. So we have never seen a double-stranded or a segmented virus with more than 12 strands, and maybe because there can only be one at each five-fold axis of symmetry. So here's the genome alignment of these viruses. Here's the double-shelled particle, and here's the core, which ends up in the cytoplasm. And again, the polymerase is thought to be inside the turret at each five-fold axis. And the RNAs are probably associated, one per turret, not lined up in the middle here. And there are, again, multiple double-stranded RNAs. They each give rise to an mRNA, which encodes a protein. So a similar strategy to the influenza virus, but of course the genome is double-stranded. Now here's a reconstruction of a reovirus, it's actually rotavirus. In the process of making RNA, these particles were allowed to make RNA, and then the structure was solved by cryo-EM, and this is one of the turrets. So on the left is the whole particle, and that's a 5-fold axis highlighted by the pink. And the RNA is actually coming out at each 5-fold axis of symmetry. And here is just one 5-fold axis. You can see the RNA threads through from the interior and comes out. So there's obviously a pore in here that allows the mRNA to come out. And as I said, each double-stranded RNA segment is attached to the polymerase, and it's attached via the cap structure of the mRNA, and the polymerase is attached to the bottom of these turrets. I think that's really remarkable. All right, the last topic is non-templated RNA synthesis. Everything I've told you so far today, you have an RNA template like this negative strand RNA, and it's used as a template for the polymerase to make copies of it as mRNAs or as genomes. There are some examples of RNA synthesis where the bases inserted are not based on a template, so it's non-templated. And this happens for the paramyxoviruses, the measles, the mumps viruses, which have unimolecular negative strand RNA genomes. These make multiple messenger RNAs, just like VSV, you can see here. But this one mRNA, the pMRNA, undergoes what we call editing to make a different mRNA that can encode a different protein. So this is non-template dependent editing. Let me show you how that works. So the polymerase is moving down the minus strand. This is just a small part of the minus strand P gene in the paramyxovirus genome. Polymerase is moving down, copying the negative strand and making a plus-stranded RNA. You can see this region happens to be kind of homopolymeric. There's lots of U's and C's. And it actually is evolved to be that way. The polymerase, as it's copying here, slips. So you can see it here. So far on the top, it's made a perfect copy. But when it reaches that C, that second C, it slips back one. So now you have a bulge. So you had two G's, and now you've slipped back. So this first G is bulging out. And then the polymerase goes back and continues copying, and the net result is that you've got an extra G in there. You should only have 3 Gs because there are 3 Cs in the template, but you have 4 because of this slippage reaction. So this is what's called non-templated RNA synthesis because there are no 4 Gs coded for there, they're just 3s. The effect is you can have a regular mRNA which doesn't have this extra G. You make one protein, but then the editing makes for a different protein, and the orange sequence shows where the protein is different. So a number of viruses, this happens in infected cells. You end up getting proteins with different sequences and different functions. So it's another way to expand the coding capacity of a small genome. This also happens in cells, it turns out. RNA editing happens in cells of various organisms. It happens in Ebola virus infected cells, and it's very important for the ability of the virus to make its glycoprotein. And remember, these are filamentous particles which are enveloped, and there's a glycoprotein in the envelope that is a transmembrane protein, and that glycoprotein attaches to the cell and allows fusion from the endosome. If editing didn't happen in the Ebola virus genome, you wouldn't get the synthesis of that protein. The glycoprotein is encoded by the glycoprotein gene, which is shown here in the genome as a red sequence. When the polymerase copies this sequence, it makes an mRNA that is not edited, and the resulting protein is secreted. It doesn't have a membrane anchor. So this comes out of the cell, and this protein is thought to contribute to cell damage. When there's an editing event going on within this gene during mRNA synthesis by a mechanism similar to what we've just told you, you now get an edited mRNA with a different reading frame, and this now encodes a membrane-bound glycoprotein. And that gets inserted into the virus. So editing is crucial for making virus particles in this case.